Okay, good afternoon, everyone. That's how I get everyone's attention. Welcome back. I hope you had a good lunch. We're back for round 2 this afternoon. We're going to kick things off with Thad Polk, and I'm going to turn it over to you.
Well, thanks. It's such a pleasure to be here. I'm going to talk about work that was both inspired by and that has basically, I think, all but 2 studies I'm going to talk about were done in collaboration with Denise. She's had an enormous impact on my career. And just a brief history to give you some idea of how big that impact is. I went back and looked at emails, and I think we started collaborating in 1999 at the University of Michigan. At the time, I was a relatively new assistant professor doing functional MRI, which was actually back then kind of a new technique. I had never done an aging study in my life. Denise had just been hired right before me, but she was, a famous aging researcher who was getting into imaging. And so we formed a collaboration, which has been the longest lasting, the most productive, and the most fun collaboration of my career. I looked it up. I think we've got somewhere between 15 and 20 publications together. We were on 3 R01s together. She was at 3 different universities across this collaboration, dozens of visits. Patty mentioned some of her most cited papers. 2 of the top 3 of my most cited papers are with Denise. And she turned me into an aging researcher. It's really her fault. So the research I'm going to talk about today is all either done with or inspired by our work together. Now, since nobody's seen this graph before, I'd like to walk through it in a little detail. But yeah, aging is obviously associated with cognitive declines in many different domains. But it's also important to keep in mind that there are very significant individual differences in age-related cognitive decline. So here's some data from the Wilson lab, where this is longitudinal data, so each line is a different person, tested about 5 years apart on some working memory and perceptual speed tasks. And so the lines that are sloped down, you can see some decline in cognition, but you also see some people who are maintaining their abilities in these domains that typically decline with age. So why do some people age a bit more gracefully than others? I want to talk about the role that inhibition might play, both behavioral inhibition and cortical inhibition in the brain. So many people in the audience will be familiar with Lynn Hasher's line of research and Cindy Lustig's work in the same area, showing significant inhibitory deficits in aging. I've listed, I'm not going to go through them, but I listed a whole bunch of different domains where you see age-related declines in specifically inhibitory function. And so as a cognitive neuroscientist, if you're interested in inhibition, you immediately think of the brain's major inhibitory neurotransmitter, which is Gaba, or gamma aminobutyric acid. Something like 40% of synapses in the brain use this inhibitory neurotransmitter. And there had been a lot of work in animals showing changes in the GABA system in aging in animals. So age-related declines in GABA release, in GABA synthesis, in neuron density, and receptor binding. But until not too long ago, it was hard to study GABA in humans. But that changed with the development of MR spectroscopy methods. So if you remember your undergraduate chemistry classes and how spectroscopy works, basically you can take a sample and break it down into different spectra, and different molecules are associated with very unique and highly reproducible frequency distribution. So you can see over here on the right a bunch of different chemicals and their signature frequency distribution that you would get from spectroscopy. And GABA happens to have its own unique and highly reproducible frequency distribution, which now you can measure in humans. So one of the, I'd say, dominant techniques now is to use a so-called megapress sequence in an MRI scanner where you collect data during an edit on phase and during an edit off phase, and then you take the difference, and that can sort of isolate the GABA signal relative to other signals that it's often confounded with. So what I want to talk about in this talk is age-related changes in GABA in humans and how those neural measures of GABA actually seem to be associated with a bunch of other neural measures that are known to decline with age and that are known to predict the individual differences in aging that we're interested in. So when you do MR spectroscopy, what you typically do is collect these spectra from what by fMRI standards are giant voxels. These are typically 2 to 3 cubic centimeters in size. So, for example, we typically collect them in 7 different voxels in the brain, and left ventral and right ventral visual cortex, left primary visual cortex, left and right auditory cortex, left and right sensory motor cortex. And then some work done by a postdoc in my lab, Molly Simonite, she actually looked at age differences in GABA measured this way in a bunch of different areas of the brain. And what you can see is that quite reliably, these measures of GABA decline with age, sort of consistent with the inhibitory deficits that are associated with aging. Now, one challenge with doing this kind of work is that, as you might imagine, GABA is concentrated in gray matter. These voxels that we collect don't just include gray matter. These are giant voxels. And furthermore, there are age differences in the composition of those voxels. So because as we age, you tend to see some atrophy, typically the percentage of the voxel that is composed of gray matter declines with age. So you might wonder whether this kind of effect could just be due to that reduction in the percentage of gray matter. But even after correcting for individual differences in gray matter percentage in the voxel, we still find very reliable age-related declines in GABA all over the brain. I think that's the only study that doesn't involve Denise directly. Okay, but now I want to talk about neural distinctiveness, which was a line of research that really was very much inspired by my collaboration with Denise. And I want to motivate this work first with a well-known animal study. So what you see here is orientation selective cells in visual cortex. Let's take a look at the left first. In a young monkey, so you record from visual cortex in a young, healthy monkey, and if you record in the right place, you'll often find cells that are orientation selective. So what you see up here in the bar chart is the response of that cell to stimuli at different orientations. And you can see it responds a lot to stimuli at 0 and 180 degrees, not so much to stimuli around 45 and 250 and so on. So it seems to be selective to these kinds of vertical bars. And you can see that graphically in this polar plot down at the bottom, where the radius from the center indicates the response of the cell to a stimulus at that orientation. So it responds a lot to the verticals, not so much to the horizontals. Interestingly, if you do the same thing in older monkeys, you typically will find that the cells are not as orientation selective. And so one way of thinking about this is that the distinctiveness of the neural receptive field here is reduced in the older monkeys compared with the younger monkeys. So Denise and I got interested in testing this in human beings. So can you find evidence that neural representations are less distinctive in older people compared with younger people? We've done a ton of different studies. I'll just describe one method that we commonly use. So using functional MRI, we'll give people two tasks that are sort of within the domain. So in this case, these are two different auditory tasks. We can do it for two different visual tasks, two different motor tasks. And in this case, people were listening to music during some blocks, and they were listening to foreign speech that they did not understand in some other blocks. So auditory input in both cases. And then we can look at the sort of reliability of the activation patterns within a condition. So it's say the correlation within the music block. So we've got three different music blocks. We can look at the pattern of activation within auditory cortex and ask how similar is that pattern of activation during different music blocks. That's a measure of within condition similarity. We can do the same thing between conditions. So we can ask, in the red arrows here, how similar are the patterns of activation in response to music compared with the patterns of activation in response to speech, okay? And by taking the difference between those two, you get one index, not the only one, but one index of the distinctiveness of the music representation and the speech representation. So when we do this in different parts of the brain, we consistently find, I think it is fair to say this is the single most reliable result I've ever found. You'll get it I guarantee, if you do this study, you will get this result. So the older adults show less distinctiveness between different categories of stimuli. That's faces and houses in visual cortex, music and speech in auditory and cortex, left and right motor presses in motor cortex. And it doesn't matter how big or small of a region you look at, the number of vertices in your region of interest, which is plotted on the x-axis, the finding is very, very consistent. And furthermore, Denise and I found that within older adults, if you measure individual differences in neural distinctiveness, it actually predicts individual differences in cognition, specifically in this composite measure of fluid processing ability. So each circle here is one older adult, age 65 or up. The horizontal axis is how distinctive the activation patterns were. I think in this case it was for faces and houses. And then the vertical axis is a standardized score of fluid processing ability. And you can see that the older people who maintain relatively distinctive neural representations tend to perform better on these cognitive tasks. What about Gaba? Well, in a more recent collaboration with Denise, we've now started using MR spectroscopy to measure Gaba in the same subjects that we measure neural distinctiveness using functional MRI. And in visual cortex, in auditory cortex, in sensory motor cortex, we have consistently found that individual differences in Gaba predict individual differences in neural distinctiveness. These studies were led by Jordan Chamberlain, who's now at Penn State, Peel Elawani, who recently graduated from my lab, and Caitlin Cassidy, who's now a postdoc with Bill Jagist. Okay, so take-home message from these studies is we consistently find neural distinctiveness, the distinctiveness of activation patterns in response to different categories of stimulus, is lower in older adults versus younger adults. And this is important behaviorally because it actually is associated with individual differences in fluid cognition, which is what typically declines with age. We also find that individual differences in Gaba predict these individual differences in neural distinctiveness. So consistent with the animal data suggesting that Gaba might play a role in neural distinctiveness, we also find that in human beings. Okay, now let me turn to a study about neural segregation. So we've heard, I think, a couple of studies about resting state functional connectivity and measures of neural segregation. And we also wondered whether GABA might be involved in that. So let me talk a little bit about that. So here's a study done here at CVL by Michaela Chan and Denise and colleagues, where they measured resting state networks and the segregation among the networks. So basically, how strongly connected or how strong are the functional connections within a functional network, and how strong are the functional connections between functional networks. And segregation is essentially a measure of the how segregated they are. So you want strong within connectivity and weak between connectivity would be high segregation. Well, in this study, Michaela and colleagues found that if you look across age, then you tend to find that within network connectivity, within these functional networks, connectivity tends to get weaker as people get older, and between network connectivity actually gets stronger. So segregation is getting reduced in both ways. So Caitlin Cassidy was interested in examining network segregation, and specifically within the somatomotor system. So what she did was she collected a battery of behavioral measures of sensory motor function. So she had a pegboard task for fine motor dexterity. She had a walking task for endurance. She had a grip strength task. And she also measured tactile function using these tactile stimulators. And then what she did was she submitted these to a factor analysis and extracted what you can sort of think of as the first principal component, or the strongest, the factor that explained the most variance in behavior. And no surprise here, the older adults show some impairments relative to young adults in their somatomotor performance. She then measured resting state brain networks. So the different colored circles here represent different functional networks based on a standard atlas in the field. And she measured within network connectivity and between network connectivity. So what you can see down here is a connectivity matrix where the squares down the diagonal are sort of functional networks, and so you can sort of see stronger connectivity within these networks, whereas the off-diagonal stuff is connectivity between different functional networks, which you can see the connections tend to be weaker. So consistent with many other authors in the literature, and I think maybe originally developed by Chan et al., she computed segregation of this sensory motor network from others. So how segregated is it? Is it very strongly within connected and not so strongly between connected, or not? And she found, consistent with Michaela's work and others, that segregation was reduced in the sensory motor network in older adults compared with the younger adults. She also measured GABA using MR spectroscopy, and as we've already seen, actually, GABA levels are also reduced in the older compared with the younger adults. And they are highly correlated. So individual differences in GABA levels predict individual differences in the degree of segregation of the sensory motor network. She also found that within older adults, segregation of the sensory motor network is good. That is, it's better for her behavioral measure of sensory motor performance. So individuals with more segregated networks performed better. GABA did too. So those with higher GABA levels also performed better on this sensory motor task, or the battery of sensory motor tasks. And then maybe most interesting of all, she performed a mediation analysis where she asked, could it be that the reason GABA is associated with sensory motor behavior is because GABA is leading to changes in segregation? So to test that, she performed this mediation analysis, First, she looked at the relationship between GABA and sensory motor performance, and then she threw in the mediator variable of segregation and found that the GABA to sensory motor performance relationship was now significantly reduced. In fact, something like 70% of the effect was explained away once you threw in the segregation as a mediator, suggesting a sort of mechanistic account where GABA is leading to changes in segregation, which in turn is leading to changes in sensory motor performance. Okay, so take-home message from this study. Like many others, resting state networks seem to be less segregated in older adults, in this case in sensory motor networks. GABA levels in those regions are also reduced, and those are related. So more segregated networks and higher GABA levels also predict better sensory motor performance, and that GABA to behavior relationship seems to be mediated by segregation. Okay, so with that, I will stop, and I'm happy to take a few questions.
I thought that's great, very nice. So I just wanted to push back a little bit on the idea that one can equate inhibition in the behavioral sense with inhibition in a neurophysiological sense. I think that is conflating two levels of explanation. And it's unfortunate they both have the same name. But just to, by way of example, within the corticostriatal loop system, cortical excitability requires greater inhibition in the direct pathway, the inferior segment of the globus pallidus. There is no simple relationship at all between inhibitory drive and even excitability or inhibition in other brain regions, let alone with respect to behavior. So I think that's, in my opinion, a step too far. It doesn't negate anything that you've shown, but I think that's maybe questionable.
Yeah, no, I appreciate that. And I apologize if I gave the impression that I was trying to make a one-to-one mapping. It was much more of an inspiration. So given the inhibitory deficits that are seen behaviorally, and given that GABA is the brain's major inhibitory neurotransmitter, we wondered if there might be a relationship. But I absolutely agree that it's not a simple one by any means.
Yeah, so that I wanted to follow up on that. And I was going to make a similar comment along the lines of, inhibition is indeed a multi-splendored and often controversial concept. But I mean, I do think that there's a way to relate them, which is, of course, like one of the major ways that you overcome interference is by the distinctiveness of representation. So there is sort of a garbage in, garbage out component to it. And I would suspect that that probably also largely explains the relationship with processing speed, right? So just like if you're going to have deteriorations in myelin, you're going to have deteriorations in speed. In this case, it's the signal itself coming in that's noisier. What I think would be really cool, actually, would be to do the similar GABA imaging on more prefrontal regions, like regions that we think are associated with either higher level representations of either the objects or in the constructs, or actually of the task representation themselves, because I think that could get into a little bit closer to what we think of as more cognitive and top-down inhibition.
Yeah, thanks. Yeah, one thing I didn't have time to get into, which I'd just like to mention very briefly, is why you might think that GABA, neural inhibition, might be related to neural distinctiveness. So if you do think about models like, there's well-known models of Duncan and Desmond, for example, in the attention literature, suggesting that the way that when we try to produce a given neural representation, it has to compete, right? And the way it competes probably is via inhibitory interneurons, which are using GABA. So you might imagine that individual differences in neural inhibition function would be related to individual differences in the distinctiveness of neural representations, and sure enough, that's what we find.
Okay. We're going to cut discussion off for now, but we'll move on because we have someone we're bringing in on Zoom, so we want to get them in on time. Yes, yes. I guess I can introduce the person that we're bringing up on Zoom while we're doing this. So next up is Richard Nisbett from the University of Michigan. Hopefully. No pressure.
Yes, I can. Okie doke. Can you see my slides? Okay. Hi, folks. Hi, Denise. I'm sorry I couldn't be there today, but I'm very glad that I'm able to participate long range. I want to talk about some research that I did with Denise. We're looking at a variety of questions related to age. Of course, Denise is the premier student of aging and cognition, but it's mostly a story there of quantitative change over time. And we wanted to look at whether there's actually qualitative change over time. That is, in particular, do people get wiser as they get older? Now, everyone seems to believe, certainly when we started our research, oh yes, everybody knows that people get wiser as they get older. But in fact, the research that was available did not show that. And the most famous research was by the Baltus group. I'm having trouble getting an advancement on my slides. So what he found was that people only get wiser up until early adulthood and not at all after that. In fact, that research and most research by others were based on samples that were not really representative of the population as a whole. In particular, they were missing people with lower educational levels. And the age groups being compared often were not really comparable. I mean, college students versus people in a center for old folks. And the materials were very brief. Sometimes only a sentence or two. There's a young girl who has to make a decision in her life, often just one sentence. Not enough cultural information, really, to allow people to work over the information and in a normal cognitive, contemplative way. So let me define wisdom just briefly. There seems to be a lot of agreement, actually across cultures and certainly within ours, that it refers to pragmatic reasoning to navigate the important challenges of social life. There always seems to be a social component to people's conception of wisdom. In our study, we looked at residents of Washtenaw County, where University of Michigan is housed. We sampled, this is in the olden days that this study was done. You could actually sample randomly from telephone directories and get in touch with people in that way. Long since not possible, at least the last 8 or 10 years. And we paid them $60 for 2 hours of study and a supplement for travel. It turns out to be very difficult to get people of lower SES and lower education into the laboratory. We had to work like mad to oversample people of that sort. But we were relatively successful in getting lots of people with low education, less than college education. Once they got to the laboratory, we gave a brief health questionnaire and a mini mental test to make sure that they were compos mentis, and a variety of cognitive tests, including fluid intelligence and crystallized intelligence. And then we asked them to read newspaper studies. In study 1, we asked them to read studies about conflicts at the societal level. For example, immigration conflicts or ethnic tensions or conflicts over natural resources. And in a subsequent study, we asked them to tell us about conflicts which they read about between siblings or friends or spouses. For these, we actually used Dear Abby columns. That is, the person tells Dear Abby what the conflict was and ask her advice. And after people read each of these stories, we would ask, what do you think will happen? Why do you think it will happen? And what do you think should be done? And we coded their answers for what we took to be wise thinking. Looking at the upper right there, first, do people seem to recognize that there are limits of their knowledge, that they don't really know enough to be clear about what they think will happen? If they applied some rule, well, I think that such and such is usually the case. Was it a rigid application, or was it one which was flexible to say, I'm not sure that I have enough evidence to apply this rule? Was there a recognition of change? Conflict situations don't necessarily remain in stasis, a particular way forever. Or was there a recognition that they might change? Did people take the perspective of more than one party to the conflict? Was there a search for conflict resolution? And was there a search for a compromise? Here would be an example of a statement that would be coded as a recognition of the limits of knowledge. Well, evidently the strong ones are going to win, but I don't know if they're democratic, are they voters, or is it going to be taken over by physical means? I don't know what's going to happen there. Here's one that indicates no recognition of limits of knowledge. But both sides are growing more vocal about change or not change. I think the next step will be basically a civil war to unfortunately solve the problem. First of all, I would say it's essential that we be able to duplicate the standard results for age and fluid versus crystallized intelligence. On the left, there's the fluid IQ results. And of course, young people are much higher in fluid IQ. But on the right, there's crystallized IQ. And as is typically the case, older people have just as much crystallized IQ skills as younger people. And here's the wisdom analyses of the social conflicts across each of these types. For each of the characterized aspects of wisdom, the older people turned out to be wiser than the younger people for these societal conflicts. And for the interpersonal conflicts, for all but one category of wisdom, the older people were deemed wiser. These are not terribly strong results, but what was strong is when we looked at the extremes. If we looked at people who are in the lower 80% of wisdom in their reasoning, you can see those are displayed there. If you look at the people who are in the highest 20%, there's a very marked effective age. Virtually all of the people who were in the top 20% in our sample were 60 or over. I'll just note, incidentally, we looked at whether there was a difference in well-being between older and younger people, and whether wisdom was correlated with that kind of well-being. And what we found was, yes, indeed, wisdom was correlated with the well-being measures that we looked at. As people get older, or at least just to compare the younger people with the older people have greater well-being. Age is associated with wise reasoning, and wise reasoning predicts well-being. Well, the other question we wanted to look at was, is there difference in wisdom across culture? We generally assume that wisdom comes with age, although until our study was done, there was very precious little evidence for that hypothesis. We know that older Americans are wiser than younger Americans in analysis of social conflicts. At least that's what we find in our research. And there's reasons to doubt, however, that this might be universally true. Cultures differ in the ways that they approach social conflicts. In particular, East Asian cultures encourage interpersonal harmony and avoidance of conflicts to a greater extent than Western cultures do. It's a watchword in Confucian cultures that you must have harmony. And effective action in our culture, Western cultures, can be carried out without necessarily cooperating with other people. But in East Asian cultures, most effective action is effective because it's carried out with other people, and you need to get along with those other people. So we suspected that East Asian cultures might be wiser in our definition of the term than Western cultures. And we wanted to see whether there's an increase in wisdom for people in Eastern cultures. So we replicated our study in Japan with a random sample from the Tokyo metropolitan area. And we had reasonably good comparable statistics demographically across the Japanese and the US population. And we found a very substantial interaction between culture and aging, such that, as I just told you, Americans seem to get wiser with age. Japanese don't get wiser with age. But young Japanese are wiser than young Americans. Now, we interpret that as saying that in East Asian cultures, you need to know how to deal with social conflict, and it's taught in the culture, and that's why young people are relatively high in it. They don't necessarily get better as they get older. Americans get better because they make mistakes and learn from their mistakes. That's all supposition, but that's what we assume is going on with Americans as compared to Japanese. So before I sign off here, Denise, I want to tell you that we also looked at whether there are differences with age in a dimension that our research group had looked at for a number of years, which is analytic versus holistic reasoning. Analytic reasoning is characterized by focusing on some object, categorizing it, applying rules, which are presumably what are operating with the category in question. And reasoning often takes the form of formal logic. East Asians are much more likely to reason in a way that we call holistic, which is to say they attend very broadly to the context. They note relationships in the broad context they're attending to. They note similarities. And there's characteristically a good deal of dialectical reasoning, which means a lot of different things. But basically, it means the same things that characterize wisdom in many ways. That is an attentiveness to the broad context that you're in and recognizing change and so on. In fact, we did not find an increase in holistic reasoning with age in either culture, which surprised us. In fact, people told us when we started to look at this, well, obviously people are going to become more holistic as they get older, partly because interdependence of a culture is related to a degree of holistic reasoning in that culture, and people's interdependence increases with old age. But in fact, we did not find a shift toward holistic reasoning. So that's one bit of research that our group did with Denise's group. And want to thank you, Denise, for being a tremendous contributor to the topic of cognition and aging, for being a wonderful collaborator, and for the luckiest of us, for being a wonderful friend.
I'm going to try and multitask here. Are there any questions?
Great talk, Dick. Thad Polk here, if you can't see me. So you pointed out that the young Americans were less wise than the young Japanese, but it also looked like in that graph that the old Americans were wiser than the old Japanese. Is that true? And if so, can you speculate on why?
Returning to that graph, yeah, that's true. And I don't really have an explanation of that. I would expect older Americans would be wiser, that older Americans would be wiser than older Japanese. That is the case, but I don't really have a good hypothesis as to why.
Very, very interesting and provocative talk. And I may think of at least some tentative hypothesis about why that would be the case, that all Japanese don't get wiser. And the case may be for what in genetics is called negative pliothropism, which meaning that there are certain genes that are very useful in the young age and then they become detrimental in the old age. So there may be some abilities, some qualities that are counter-wise in young age, but then they serve people better if they preserve them in the old age. So that may be sort of a case of cultural pleiotropism, when in Japanese culture, according to your studies, maybe young people already sort of are inculcated with those trappings of wisdom, and they keep it for the whole life. But in Western culture, young people are more exploratory, more searching, they made more mistakes, they do more stupid things, but eventually they acquire some baggage that helps them in the later life. Just
That's
a
a very
speculation.
Very interesting perspective on this, on these findings.
I think we have time for another question. Okay.
Dick, thank you for a wonderful talk. And one thing I think about aging and culture is that wisdom is the ability to overcome the constraints of your culture, in a sense. So you would expect analytic people, if you're always analytic, you make X number of mistakes by being analytic when you should be holistic. If you're always holistic, you make X number of mistakes by always being holistic. So I think maybe a switch over from one culture, but that in general you don't show such a strong preference for analytic or holistic thinking, depending on which culture you're from, because you learn that by constantly applying the same rules, you make mistakes, and that you learn flexibility, and that's what the essence of wisdom is.
Right. Yeah. Very good.
One last question.
So this is Cindy Lustig. Although it is the data that you showed were cross-sectional, I found it really interesting that the mean difference between the young and the old was really driven by a select group of older adults. And so I wonder, and it seems like the big thing from sort of a societal point of view is how do we identify those young adults or create the conditions for those young adults to become those more wise older adults. And so have there been longitudinal studies either sort of looking at trajectories or even better identifying some of the factors that might contribute to those?
I'm not aware of longitudinal studies. I should say there's been research, this research was published several years ago. There have been a few studies since, only some of which find that people get wiser with older age. I don't think anybody has done as good a job of demographically, of getting a substantial segment of people with relatively low education across all of the age groups. But you put your finger on something that we're missing. I don't believe anybody has done the longitudinal study, and we're not going to really be able to make conclusions about whether people get wiser with age, I think, without seeing what happens as to an individual person as that person gets older. Because that controls for everything that we don't control for in these cross-sectional studies.
All right, thank you so much. Let's have another round of applause. And next up we have Michelle Carlson from Johns Hopkins University.
Oh, I hear it, yeah.
That's definitely on.
Yeah, that is on. So now for something a little different. I'm a translational researcher. I work in the School of Public Health, as you see. So I carry my background. I can't possibly not represent cognitive aging, but what I do, and I hope we'll keep time on this, but and my lab website, I encourage you to go to the lab website because I won't get to tell you everything about our lab, including our wearables work, that I'd love to do some other time. What I want to do is make a moment here just to talk a little bit about the vision that Denise really brings to my work. And so I haven't worked directly with Denise, but the work that she has done has been very influential to my work and kind of emboldening me as well. And so, for example, I'm very interested in real-world engagement. How do we bring the real world, how do we train to the real world, essentially? If cognitive training trains to that particular outcome, if it leads to near transfer to whatever we're training, well, let's train to what we want to improve in daily life. So that's my mantra. And I thought the Synapse trial that Denise led was fantastic. And I was amazed it got, congratulations to NIA for funding it, because at that time, those kinds of trials were kind of not really the zeitgeist. And so her work really suggested that purposeful engagement and something meaningful probably had as much value as the training that we were doing up to that point. Another premise is neuroplasticity occurs throughout the life course. It doesn't start or stop anywhere in the life course. So the scaffolding theory, thank you to Patty for sharing this morning or this afternoon. I think that this idea applies to my thinking as well. If we're going to think about neurocognition, it can be manipulated and it can be measured in a translational setting. And the environment is an important third rail. So I'll be talking a little bit about the environment as it relates to intervention placement. When you're thinking about an intervention, don't think just about the individual. Think about that context and the life space of that individual because they're interacting all the time. So we provide, in doing intervention research, providing neighborhood infrastructure for safe engagement is important. And actually, I think the Synapse trial, what inspired me was you actually said you rented out a structure in a strip mall. Yeah, I was like, that's awesome, and a lot less expensive, too. So just some really, yeah. So to be quick, I do want to bring about this idea of understanding the role of enriching activity throughout the life course. We typically think about the life course as a period of getting stale and getting worse and aggregating chronic diseases, and that's the way we should think about it, because that's the medical model I live in and most of us live in. But if we could kind of reverse this, as was mentioned earlier by Richard with Denise, is this idea of kind of flipping the equation a bit. What gets better with age? And so I start, I'm going to get to that, but as I start, I want to talk about some of my students' work over the last couple of years. And some low-hanging fruit is looking at what do people do when they're young, and does it matter in late life? So I'm going to apply the retrospectoscope and say, well, let's take a long-term brain print, so to speak, and let's apply a really simple questionnaire to look at early life activities and the kinds of activities people will remember that they did or didn't do. So we did this in a study called Experience Core at baseline in 700 adults. And we asked them, yes or no, did you do any one of these 7 activities? And we just tallied them. And then we looked at them in relation to late life, basically up to 40 to 50 years later. We looked at their relationship to both matrices of structural brain health, and we also looked at them in relation to behavior, cognition. And in short, this reflects 2 different papers, one led by Kyle Morid and one led by Thomas Chan. And in both papers, they showed that there was a direct correlation between the tally of these early life activities and these markers of brain health and measures of functional, of cognition. And that there was actually a functional correlation for the brain imaging data. The larger your hippocampus, the better your memory. Now, these are cross-sectional, but what was important about them is that they controlled for other late life measures. So there was still variance to be accounted for with this early life questionnaire, even after you accounted for a host of medical comorbidities, late life activity, physical activity, using something called the CHAMPS, et cetera. So that was kind of exciting. Going from there, my student, Brianna Crane, was inspired to go to the Health and Retirement Survey, which is a much larger study, and they recently added a questionnaire a little bit larger because they were looking at adolescents. And with adolescents, they could survey more activities. And what she did is looked at a similar tally in relation to late life cognitive health. and which she found in unadjusted models and then fully adjusted models. It was that for each additional tally, each point that additional activity you endorsed, you had a 0.6 point increase in a global cognitive score. And that finding was significant. So these results are consistent for both early life and adolescence in suggesting that there may be some value to looking at capturing early life as still being sort of aggregate and helping with late life function. So that leads me to this slide that many of you see all the time, is this focus on, we've spent a lot of time on amyloid deposition and tauopathy as far as how do we target it, not so much tau, but amyloid. What we're learning, and I think Nick mentioned this morning, is this idea that we're kind of looking, or maybe last night, that we're looking at now kind of what can we tell people about how to modify their risk. And a lot of the factors that we thought were good for us turn out to be good for us based on a number of important studies, and most importantly, or the most comprehensive, is led by Livingston et al. In 2020. It shows that you can account for 40% of your risk, or 40% of your risk is potentially modifiable. And the most recent iteration, air pollution and sleep, were added. So the body of modifiable risk factor grows. And I provide a link for you. I participate with the AARP on a Global Brain Health Council, and they have 7 pillars of health. And it's really a good evidence-based tool if you want to pop on there and look at any one of those 7 pillars. So now, in terms of thinking what to share today, I know I wanted to talk a little about sort of the reason why I like to focus on translational interventions, and among them is this idea that we know physical activity is good for us, but we don't get it. And why not? Why don't we get as much physical activity in daily life as we need? Well, one is possibly that I'll come to, but I think the first question is just a little enough? Are small amounts of activity sufficient to get people out of their homes, out of bed, just moving around, because there's so many sedentary adults out there. And you've heard sedentary sitting is the new smoking based on an epidemiologic study. So in our world, thinking, how can we actually kind of get people from a sitting position to a standing position, and does it matter? So this study, 2 different studies here, provide proof of concept that if we use looking at hippocampal volume, that step activity monitors, just wearing something around the ankle to capture your weekly step activity, is positively correlated with hippocampal volume. And these step times are fairly low. They're not people who are running, or really, most of their activities in a low intensity range, meaning under 100 steps per minute. The people on the high end are male men. They're retired male people. So these mail carriers, it seems like their career occupation served them well, because they continue to walk around quite a lot. But what we also found next among people who are mostly walking a lot less is that physical activity is not just associated with hippocampal volume, it's associated with a subregion of the brain, of the hippocampus, called the subiculum. And why is the subiculum important? Well, functionally, it appears to be functionally related to spatial navigation. And it's also important as an early sign for Alzheimer's disease. So there are kind of clinical reasons and also functional reasons to think about modeling the hippocampus in response to environmental enrichment. And again, I get back to this question, how do we get people just to be more active? Because we know it's good for us. We know that it's probably not likely to get many at-risk people to do 30 minutes of vigorous or even moderate intensity activity a day. And we know that a lot of people live in environments that really preclude that safe kind of activity. So this is where I focus on this idea that, I might have taken that slide out, is to look at a program that really was developed over, I'd say, 25 years ago, called Experience Core. And over a series of kind of feasibility studies and pilot trials, and then a funded trial by the NIA, we've been looking at the idea that volunteering is actually potentially good for you, because it harnesses your cognitive activity, it harnesses physical activity to get up, get out of bed, go to an elementary school, and work in teams to help children with who are in need of, who are in need of literacy support. So what Experience Core volunteers do in teams, and we did this trial in Baltimore City, was to look at giving them the tools they needed over like a 1-week training period, And then in teams, they would work with an elementary school teacher in grades K through 3 and basically support them in book selection, the library support. They might actually also do some eye-to-eye positive behavioral communications and essentially do anything that the teacher needed to help with the kids who needed just a little bit more attention. And it helped that teacher give more attention to other kids in the classroom. So we conducted that volunteer program over a 2-year trial and then looked at multiple outcomes: physical function, cognition, psychosocial well-being, and we did a nested brain health study where we also included physical activity measures. What was important about this trial, and I think I pulled the slide out, so I'm going to jump to it real quick, is that if you look at this image of this older volunteer with the children, how can you deny that they are actively engaged with one another, and how could you deny that probably isn't good for both entities? So I think this gets back to the slide I took out, which was wisdom. Probably it's a great feed off from Richard's talk, because I will posit and propose we have data to support the idea that you can engender wisdom in aging adults, but it may be that what happens is people don't feel they have anything to give. And so what we did is we gave these older adults sitting in their communities, knowing that their communities were in great need, but not knowing how to serve them. We gave them an infrastructure and a set of tools, and we empowered them. And the schools love them. They want them desperately. But what happened is, I think that to answer maybe part of the question from the prior talk, maybe one doesn't know they have the wisdom to give unless they're given an opportunity to give it. And in this model, it's possible that these children should be followed. It may be that you just designed a study that the younger people who were part of this program they had an intergenerational exchange that may have actually helped the young people engender wisdom in a way they might not have otherwise done. So I know we don't have a lot of time I want to share another intervention, but this is really clearly a real-world intervention and engagement. And the people that were in this trial were really at elevated sociodemographic risk by any metric: health metric, race, income, body mass index, mini mental score, gender. So they had a multitude of risk factors that would place them at greater risk for cognitive and health deficits. And across a number of papers, which I just, in the interest of time, summarized here, we showed modest benefits across all of those outcomes, but they were modest. And sometimes they were more gender-oriented. So in some cases, for example, in the brain health study, we showed that there were functional benefits in hippocampal volume. There were improvements in hippocampal volume for both men and women, but the men showed us more improvement than the women, and we don't know why. So why would male volunteers benefit more than female volunteers? We didn't. We have speculations, but we did not a priori propose that. We did see functional correlations in both men and women who volunteered. The larger hippocampal volumes were related to, or increased hippocampal volumes over the 2 years were related to improved memory. So there's a functional correlation. And kind of moving from there now, I'm kind of moving into this area of the amygdala as an important region that's also highly plastic and highly related to sociocultural factors and a need, sort of more emotive needs. And so it could be a key hub and a biomarker for volunteer-related changes. And so we've got some preliminary data, well, it's under review right now, looking at whether or not we saw some changes in the shape of the amygdala in the volunteers versus the controls, and there are some promising results there. So the results suggest that you can enrich and directly measure both cognitive, psychosocial, and brain biomarkers related to this enrichment. The challenge is scaling up, for example, and justifying its scaling up, because it is somewhat resource intensive, and you need schools that can afford and absorb this kind of an activity. And so school infrastructure is an issue. There is an age bias that we confronted and had to address. And there's also COVID restrictions as of late that were a little bit of a challenge. So our next steps with funding from NIA are to do a long-term follow-up to see if those short-term benefits of the trial accrued to lower long-term risks and costs related to dementia, disability, and frailty. And so we're using Medicare data, Maryland CRISP data, National Death Index data to look at these questions. And we're actively doing that right now. If anyone's interested, it's a wonderful data set. And as I kind of move to the next part of thinking about enrichment, I mentioned environment. And environment is so important, providing the structure and a place where they can come and be enriched and active is so important when we look at using census data, for example, to socio-demographically characterize the participants in the Experience Corps trial in Baltimore City. You see they come from a number of different locations across the city, and the color coding is to show you that they are actually the darker, the more orange the area, the more socioeconomically deprived they are. So what that suggests to us is that there may be a relationship between participating in the intervention and the level of benefit they may get from having been in a structural setting. Clearly, physical activity in the orange neighborhoods is harder to get than in the lighter colored neighborhoods. We're also looking at environmental factors that may affect outdoor engagement. And it turns out that in cities, urban areas are also at greater risk for air pollution, especially when you have urban canopies that trap the air. So we're looking at that as well. And we're doing that through another study that I'm just going to basically mention. We're actively looking at a number of environmental risk factors in an observational study that spans 30 years with the goal of trying to identify whether or not physical activity, cognitive activity, and other environmental risk factors might either enhance or mitigate the rates of cognitive aging and dementia in this large-scale study. And after that, I end with this idea of providing a safe environment. How do you, if you can't get the environments, the people to the environments, then you bring the environment to the people. So this is the last thing. I'm spending a lot of my energy on this. This is the idea of simulating real-world engagement in communities in basically as close as a movie room. And this is called the Optimizing Cognitive, Environmental, and Neuromotor Stimulation. Obviously, I wasn't trying to pander, to get oceans out of it, so such a simple title. So what it does is we're taking an intervention, a 3D gaming system, pretty sophisticated, and you play it standing up, and essentially the goal is you interact, you have a little joystick, and this arm navigates Bandit the dolphin in three-dimensional space. And what you're doing is you're exercising all levels. You're cognitively stimulated, you're physically stimulated, and you're very much socially stimulated in this underwater game that was initially developed for stroke rehabilitation. So yeah, I might do one extra, but so this is a session. I may not have time to show you the set. Well, here's Mr. O after, and he is going to town. They love this game. And this is in a movie room at an independent living community. So retirement communities are really excited about this. And we were able, he has Parkinson's disease, and this is his first session, his latter session, and he doesn't want to stop, and he mastered the game. What we learned by implementing this is that we have metrics we can apply. Even in this simulated environment, we can look at learning over time. And what we see is that every participant in this particular feasibility study learned. And we can see how well they get better at the game. And as they get better, the challenge increases. The fish get faster, the sharks come along, and sharks are not nice, and they nip at Bandit, and the participants want to protect Bandit. So if you see this, I just want you to see, I don't know if you'll see Mr. O getting attacked by a shark, but he wants to make sure Bandit does not get bitten by that shark. And so he's really motivated, and every participant's motivated. So what we're looking at here in this feasibility study is that you can correlate their improvement over trials and then relate that to metrics of pre and post cognition. And we did see some correlations between their mini mental score pre and post as a function of how quickly they learn the game. So we're now doing this as part of a Department of Defense grant in older adults with past TBI because they're at greater risk for dementia. And so we're doing the same kind of idea, and we implement wearables at baseline to see whether or not we can see if their life space out in the environment is changed as a function of playing this simulated real-world game. Will they actually show more activity outdoors? So that's kind of where we are. And I just want to show you how socially they really love Bandit. And so what we did is we said, would you like a picture with Bandit? We bought this life-size dolphin, and they put that on their fridge, and they show it to their friends and their family. So if you didn't think it was social, believe me, it's social. And these people are varying levels of physical disability. They would not do a walking exercise intervention, guaranteed. So I want to thank my team. It takes a village, and this is my team on many different levels. And I think sometimes you just start small and then use biomarkers and various, really trust that purpose is really important, and And then I'll leave it with that.
We have time for a few questions.
Hi, great talk, Michelle. I was wondering, what's your thoughts on the maintenance of these effects with the experience corps? I mean, I love that intervention, and we see from other intervention work that, you know, once you seize the ability, then the effect on you know, changes in brain structure, you know, wheels off. And so do you think that an intervention that just targets just not one ability, but, you know, social aspects and cognitive aspects, that will help the maintenance effect?
I think you are anticipating my answer, which is that, yeah, durability is the question. And, you know, if you look at other studies like the juggling study or a college exam, we see that the benefits, you know, they regress to their prior state. Here, we think that we were actually changing their lifestyle. I think that's kind of partly how you asked the question, is that we gave them a group of a social network. And I was curious with Denise whether that happened in the Synapse trial. Yeah, that'd be an awesome study to do is see follow-up. Right. That's so important. I feel like what happened and what probably happened in Synapse is something greater than we could have designed.
Yeah, we could not what happened, but it was really dramatic. The effects of people who come back, and they also maintain the behavior, so that they can, not all of them, but a subset continue to quilt. One woman said she learned that she could learn, and it made her take up painting, that she didn't like quilting, but she loved that she could learn to learn. And the effects were unbelievable, and even though I wasn't successful in getting further funding through study sections, I think it was probably the most important study and had the biggest effects on people that I've ever done. It felt really good.
That's so well said. I can't improve on that. Yeah, and I think with Experience Corps, they would go gambling together. I don't know if it's quite as common. They get a bus and go to Atlantic City, and yeah, or well, actually Maryland has gambling casinos everywhere. But anyway, yeah, we did things we didn't expect to do. So to answer the question, Jared, I think we're going to expect in the long-term follow-up, we're actually trying to get telephone interviews, and it's been hard because it's like cold calling. So we have to regain the trust that we've had. But I think what we'd like to know is whether or not they do show any long-term maintenance of their physical activity. Yes.
All right, thank you.